Alrighty, hello everyone. In this video, what we're going to be talking about is really one of the techniques that involves nucleic acid sequencing. So if we wanted to sequence a DNA molecule, what we're trying to do is to determine the exact, precise sequence of nucleotides in a DNA sample. One method is known as the chain termination method, or as the dideoxy method. It is also named after its founder, Frederick Sanger, which is known now as the Sanger method, right? He also won the Nobel Prize for it in chemistry in 1980 for his achievement. All right, so what exactly is the chain termination method? If we think about nucleotides, we had said that they are connected to one another via a phosphodiester bond, right? Between the phosphate group at the 5' position and the 3' position hydroxide group, right? So what happens in the chain termination method is that incoming nucleotides are being added as deoxynucleotide triphosphate. So what you have in solution is a deoxy CTP, a deoxy TTP, a deoxy ATP, and a deoxy GTP. All right. And the nucleotides are being added to the 3' end, to the free hydroxide, forming a phosphodiester bond. And that particular reaction is catalyzed by an enzyme known as DNA polymerase, right? And synthesis takes place from the 5' to the 3' position. So if we have a DNA of interest, which we'll call our template DNA, what we'll need to initiate the addition of incoming nucleotide is to add a primer, right? And primers will always be 5' primers, right? You have the primer, it's few nucleotides long, it will anneal to the 3' end of the template strand, right? So you have the primer, you have 3' end of the template, right? Template strands. And then what you do is you add incoming nucleotides, right? And to the primer, a phosphodiester bond is formed to incoming deoxynucleotide triphosphate, including ATP, CTP, and GTP. And what catalyzes the reaction is the enzyme known as DNA polymerase, right? So now all 4 nucleotides are present in solution. The next nucleotide in the template strand is G, so it will pair with C, so CTP or deoxy CTP will add, form a phosphodiester bond, it will base pair with G. The same case happens up until the entire sequence. The complementary sequence of the template strand has been synthesized. So, and the enzyme is known as DNA polymerase. So we're going to talk about 2 different ones. One when we're actually running it in a lab in a smaller scale, and one that is more of an automated method. What exactly is taking place? But again, we have a template strand, and that template strand is what we are interested in finding the sequence of, all right? So we don't know what the sequence is of the template strand, right? You have a DNA sample and you're interested in figuring out the exact precise sequence of its nucleotide. So what you do is you add a primer, and the primer and it's short, few nucleotides long. It's a 5' primer. It will anneal to a 3' end of the template strand, right? So somewhere within the strand, but it's a 5' primer. And then what you'll need is a primer to begin with, and the DNA sample, or the DNA template that you're interested in sequencing, determining the exact precise sequence of. And then we add DNA polymerase, and the function of it is, again, to catalyze the addition of incoming nucleotides. So what you'll also need is all 4 nucleotide or deoxynucleotide triphosphates, DATP, DTTP, DCTP, and DTTP. Now notice the name of the method is known as the chain termination method, right? So suppose you wanted to keep adding incoming nucleotide. What is required is that 5' is that 3' hydroxide group, right? Because that's where the the phosphodiester bond is formed. So if it lacks, all right, if you end up having dideoxynucleotides, which again lack that 3' hydroxide group on the sugar, then synthesis will basically terminate, right? DNA polymerase will no longer be able to add or catalyze the addition of the incoming nucleotide because it requires that free 3' hydroxide group. So it will terminate, and that's why we call it the chain termination method. Meaning, if you have in solution deoxy-ATP, deoxy-CTP, deoxy-GTP, and deoxy-TTP, and you go in as well and you add the dideoxynucleotide triphosphate of each one of those, there's the competition where DATP might add, or a dideoxy-ATP might add. If DATP adds, then synthesis will continue, but instead of the dideoxy-ATP adds, synthesis will basically terminate at that point. And what we typically do is we add more of the deoxynucleotide triphosphate as compared to the dideoxynucleotide triphosphate. So now, suppose you go in and you incubate that particular DNA template with a primer, DNA polymerase, the deoxynucleotide triphosphate, and a dideoxynucleotide triphosphate. Well, in the automated method, what we do is we add a fluorescent labeled to each of the dideoxynucleotides, and we add a different color for each of the dideoxynucleotides, and we'll see in a moment why. But now let's look at the number of different fragments that will actually generate from that particular method, right? So the primer adds to the template, and it will basically anneal to it, it'll hydrogen bond, and then if you take a look, the next nucleic acid in sequence is basically G. So I'm just gonna go ahead and write it. CGG, TAG, CAACT, right? And then we have the 3' ends. All right, now our primer is GC. Just gonna keep it, that's our primer, right? And then what we have in solution, all 4 deoxynucleotides and all 4 dideoxynucleotides. So now the next nucleotide in sequence is G on the template strand. So its complementary is C, right? If deoxy-CTP adds, right? If deoxy-CTP adds, synthesis will continue, then the next one comes. And if A was deoxy-ATP, then synthesis will continue. You'll get T, and then if what adds is deoxy-CTP again, you'll get C, you'll get G, you'll get T, T, G, T. A, right? Well, what if instead at that particular nucleotides, instead of the deoxy-CTP adds, which has the 3' hydroxide, a dideoxy-CTP adds. Well, what happens is a dideoxy-CTP will cause it to terminate. It lacks the 3' hydroxide, so synthesis cannot continue, right? So we'll get 1 fragment, but because what we put in solution is more of the deoxynucleotides than the dideoxynucleotides, we end up getting all different fragments. And each one that has, this fluorescent labeled di at the end indicates basically what is the last nucleotide that is present in that particular sequence. And then all the fragments will pass through a capillary gel that will separate them based on size. If you recall what we talked about in gel electrophoresis, right? It's separating the molecule based on size, where the smallest fragments, the shortest fragments, will migrate farther down on the gel, right? All right, so they'll pass through a capillary gel, and they're labeled with a different colored dye. They'll pass through a laser, a detector, and then what we get is the exact precise sequence, right? They're separated based on size, so we can know which one is the shortest, and then the next, and then the next, and we can determine the exact sequence of a polynucleotide. So here's another example where you're actually looking at the bands that would come on the gel, right? The shortest band, the shortest fragment will be farther down on the gel. And if you know which dideoxynucleotide, so in this case, what it's telling you that on this lane, we added everything that had, or we incubated the nucleotides with dideoxy GTP with all 4-deoxynucleotide triphosphate. On this lane, we incubated it with dideoxy CTP and all 4-deoxynucleotide triphosphate. So every time we see a band on the gel, we know that that particular band is a C in the sequence, right? So we get A, T, A, etc. But this is reading it 5' to 3'. And if we're interested in the template sequence, remember this is complementary to the template sequence. So the actual template sequence is complementary to what we see on the gel. It would be T, A, C, C, A, etc., right? What we see on the gel is the complementary sequence of the template strand, right? Because what we're synthesizing is the complementary sequence of the template, and we're looking at it 5' to 3' direction. And its complementary strand of what we see on the gel is the actual template sequence. That's something to keep in mind. What else would be missing on the gel, right? What else would be missing on the gel? What won't separate on a single 2 bands? It's the primer, right? The primer is not included on the gel either. It will be included with the very first nucleotides, right? It won't break it up into 2 nucleotides. So now let's consider an example. All right, so suppose you wanted to run it in a smaller case in the lab. What you do is you have a DNA of interest, and that's what we call, again, our template DNA. And then we add a few nucleotide primer. And what we need to keep in mind is that the primer is a 3' primer. And then we separate it into 4 different solutions, right? So this is solution number 1. And what we put in solution number 1, I'm just going to make it into a cuvette. What we put in here is our DNA polymerase, which will catalyze the addition of incoming nucleotides. We will also put in our DNA template. That's the one that we're trying to figure out the sequence of, right? We don't know the sequence of the DNA template. That's what we're trying to figure out. We will also put all 4-deoxynucleotide triphosphates. So we'll put DATP, deoxy-ATP, deoxy-TTP, deoxy-GTP, and deoxy-CTP, right? What I typically do, instead of writing all, I'll just put deoxynucleotide triphosphate, right? All 4-deoxynucleotide triphosphates, all right. But if that's all we put, we'll get one long complementary polynucleotide. So what we end up doing is, for one of those 4, we'll end up putting a dideoxynucleotide, right? So suppose in sample 1, right, and that will be sample 1, right? In sample 1, it'll be lane A, right? So I'm going to add dideoxy ATP to it, right? So what happens now, if you have the sequence, is the follows. I just need a pen. Okay, so the sequence is 3', So C, C, G, G, tag, C, A, C, T, 5', right? That's our template, right? That's the template. That's the sequence we are trying to figure out what it is. Okay, this pen works better. All right, and then we have our primer. Primers are 5' primers, and in this case, there are 2 nucleotides long, so GG, right? So in sample 1, what we have is a dideoxy ATP. So every time we have the complementary to, T pairs with A, right? So every time you have a T on the template strand, A will pair with it. And then in this case, you have 2 options. It could either add the deoxy ATP or the deoxy ATP. Okay, so G, C, we have a C, so here's a C. G, C, we have a C, and then we have a T. Now it has 2 options. If a deoxy ATP adds, right, I'll just make it in pink again. It will terminate, it will stop. It lacks the 3' hydroxide. But remember, you don't have one template in solution, you have many, right? So if instead it was a deoxy ATP, then it will continue. You'll have C, C, A, T, C, G, T, T, G. And then the very last one, whether it's the dideoxy ATP or whether it's the deoxy ATP, it wouldn't really matter because it will terminate. It's the end of the sequence. So you end up getting 2 different fragments, right? You end up getting 2 different fragments. And if you put those fragments on the gel, they're separating based on size, right? The shorter fragments will migrate farther down, the longer fragments will migrate a bit slower. And if you do the same in the presence of a dideoxy CTP, right, then every time a C is to add, it will basically terminate. So let's try it. Right? So now suppose we are using a different sample. Instead of having the dideoxy ATP, we have a dideoxy CTP. So now every time a CTP is to add, or a deoxy CTP, it has 2 options, a dideoxy CTP or a deoxy CTP. If a dideoxy CTP adds, it will terminate and we'll get a fragment. If the deoxy CTP adds, it will continue on. If, again, a dideoxy CTP adds next, it will terminate. So suppose a deoxy CTP adds, then it will continue on. T, C. Well, if a dideoxy CTP adds, it will terminate and you'll get this fragment. But if instead a deoxy CTP adds, then it will continue on, right? And then we have T, T, and then you have another G, and then a T. And then you end up getting 1, 2, 3, 4 fragments. Notice you will get the longest fragment, right? So in this line, we end up getting 1, 2, 3 fragments, but we will, I don't know if you see it, we will get that longest fragment again, right? So how do we know what is the last nucleotide in sequence? The one that has the dideoxy and the deoxy would be slightly a thicker band, right? But really all of them will show a faint band. This band will just simply be thicker. All right, and you do the same for the next 2, right? And then each one is placed in on a different lane on the gel, right? And you let the gel run. The shortest fragments will migrate farther down on the gel, right? And then you know which dideoxy nucleotide was present in each of the lanes. So that's what this is labeled. It's labeled based on which dideoxy nucleotide is present. Okay, and then you go from the shortest one, it is C, and then a C, right? You have a C, and then a C, and then an A, and then a T, and then a C, and then a G, and then a T, a T, a G, and A. Right? So we have the sequence, but again, that's the sequence of the complementary strand to the template, right? And if we're really interested in the actual template strand, it is complementary to this one, right? So it goes 5' to 3'. It would be T, C, A, G, C, G, A, T, G, G, right? But it is missing the primer, right? Plus the primer. But typically we would know what the sequence of the primer is, right? And in this case it is GG, right? Notice you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The sequence is 10 plus the primer, right? We don't see the primer separating on the gel. Okay, well, I hope this was helpful. We will do some examples of those in class later on.